Hello students, welcome back to this video session of Pharmaceutical Organic Chemistry 1 Practicals. In this video session, we are going to discuss an important concept, that is derivative preparation. So it is a preparation of suitable solid derivative from the given organic compound. Now we are discussing about the systematic qualitative analysis of unknown organic compounds. In the previous videos, we have discussed a lot about how we can identify the given unknown organic compound using a systematic qualitative analysis approach. We have seen when the solid or liquid compound is given to you and you are supposed to identify what the compound is, you have to perform a series of tests which are referred as the qualitative test. So we should start with the preliminary test, then we go for the detection of elements which are present in the given unknown compound other than the carbon and hydrogen. Then we perform a solubility test to categorize our compound into group 1 to group 7, and then we subject it for the suitable functional group test, and then we can find out the melting point or the boiling point of the compound. Sometimes what melting point or boiling point we are getting, they are not actually guiding us to certainly identify the given organic compound. Okay, so there is no much surety about the identity of the compound due to the melting point or boiling point which we are getting, that is not very sharp. So what we can do, we can prepare the derivative of that unknown organic compound, and then the derivatives will give a very sharp melting point because they are quite pure than the given unknown organic compound. So there is a need to prepare the derivative of compound which you want to identify, and then it will be very easy because it will give a sharp melting point. So we'll see how how the derivative can be prepared and how it will help you to identify the given compound. So the aim of this experiment is to prepare a suitable solid derivative from the given unknown organic compound and then confirm the compound by finding out its melting point. Okay, so we'll see what is a theory or principle behind this particular derivative preparation. So we have seen the preliminary examination and group classification test. They indicate the particular class or functional group to which the unknown organic compound will belong. Okay, so it is always better to identify the unknown compound with certainty when it is converted to the sharp melting point solid compound. Okay, so what we have to do, when we have a solid compound which is not giving a sharp melting point, we can chemically convert the solid compound into another solid derivative, okay, which will have a sharp melting point. If you have a liquid compound, we will try to convert into a solid derivative, and then solid derivative will give a sharp melting point because it is more pure than the crude compound. And once you get a sharp melting point, then it is always easier to find out the identity of the compound. So this conversion of solid or liquid compound into its sharp melting point solid compound is referred as the derivative, and the procedure what we follow, we refer it as a derivative preparation procedure. Okay, so these liquid derivatives usually they are not considered suitable, again because they will not give a sharp boiling point. Okay, so we are not able to identify with certainty that the given compound is so and so. So what we can do, we can convert the liquid compound into a suitable solid derivative which will have a very sharp melting point. That is the idea behind forming a derivative of your solid or liquid compound to identify the unknown compound. Okay, so when we are converting our compound into its derivative, we have to see to that the derivative should be prepared in such a way that it should at least melt above 80 degrees Celsius. It should not have a melting point below 80 degree, and this should be kept in mind. And it is always better, whatever derivative you are preparing, either of solid or liquid, it should have the melting point between 80 to 150 degrees Celsius. So this is ideal melting point for the derivatives which we are getting in a solid form. The compound is identified by cross reference of its melting point or boiling point with the melting point of its derivative. Okay, so this procedure we can utilize and we can qualitatively identify the unknown compound. When preparing a derivative, it is necessary that the product be obtained in a pure form, because unless and until you are not having a pure solid derivative, then it will not give a sharp melting point. We have discussed in the previous video about determination of melting point. There we have discussed when we get a sharp melting point and in which cases we get a melting point in a range. Okay, so it starts at a particular temperature and within 1 or 2 degrees Celsius it will completely melt. So in such case we can easily say the compound is not pure. When compound is highly pure, like the derivatives what we are getting after recrystallization, they are giving sharp melting point. Okay, so the idea behind or intention behind converting our liquid or solid compound into its suitable solid derivative is for identification. Okay, so identification is only possible when you have a pure solid derivative, and then only it will give the sharp melting point, and that will actually help us to reveal the identity of the unknown compound. Okay, so purity is very important for the derivative what we are going to prepare. And for purification, once we do a chemical conversion, once we do a chemical reaction, the derivative what we are getting, it may be a crude derivative. So what we need to do, we need to follow some purification procedure, and one of the simplest and widely used purification process is the recrystallization. Okay, so you might have studied or heard about the recrystallization. So recrystallization is a process by which we are converting the crude compound into a pure compound or pure crystals. Okay, so it involves a series of steps. Recrystallization is very important to get a pure compound, and it should be done very carefully. So what you have now the crude derivative, and this crude derivative should be converted into a pure derivative. So it involves a series of steps. So what we do first, we take a crude derivative and we dissolve this crude derivative in a suitable solvent. Okay, so you have to select the solvent in such a way that the solvent will have a very high solubility towards your solid derivative. Okay, so you can heat that solvent, and while it is hot, why you are going to heat the solvent? Because the solvent should dissolve the crude derivative. Okay, and the impurities, if at all it is present in the crude derivative, that will remain insoluble. Okay, so what you have to do, you have to dissolve your crude derivative in hot solvent, and then while it is hot, you have to filter this through the Buckner funnel. So this is a Buckner funnel. You can put a suitable filter paper, and while it is hot you can filter it. Okay, you can filter it even without the suction. Then in the filtrate, that is in the bottom beaker, you will get a solvent plus your solid derivative which is in a dissolved form, and the impurities will be retained on the filter paper. Okay, so you can wash this filter paper, okay, with some residues with the cold water. So if at all any crude derivative is there, that will retain on the filter paper, and the pure crude derivative will go into the filtrate, right? And then what you can do, after washing, you can get the beaker with the filtrate, and then you can cool it. So once the solvent will evaporate, what will happen? The solid derivative will try to crystallize out, and then it will form its pure crystals, and then you can evaporate the solvent slowly and you will get a pure recrystallized product of your solid derivative. Sometimes what happens during the chemical conversion, the colored impurities are also getting formed, and these colored impurities will make your derivative in a colored way. So you can remove these colored impurities by using the activated charcoal. So activated charcoal is the powder which looks like this, and this actually help us to remove the colored impurities by the principle of adsorption. So what you can do, you can take your crude compound of solid derivative and you can dissolve in a suitable solvent. You can boil it, you can heat it, and while it is hot you can add a pinch of activated charcoal. Okay, and then you can stir it. So you can boil this for about 5 minutes, and while it is hot again you have to filter through the Buckner funnel without suction, and the filtrate will contain the derivative which is now free from the colored impurities. So what will happen, the colored impurities will adsorb on the activated charcoal because it will provide a high surface area, and that will retain on the filter paper. Okay, so this is very popular method, and you can see in the figure the bluish crude derivative was converted into a sky blue color pure derivative. Okay, so this dark blue color are nothing but the colored impurities. So what we have done, we have taken these colored impurities along with the crude compound and we have dissolved in a hot solvent, and then we have added activated charcoal, we have boiled it for 5 minutes, and then we have filtered it, and in the filtrate after evaporating the solvent we got a pure solid derivative. Okay, so this is how we can get the pure solid derivative from the crude derivative by utilizing a proper solvent which can selectively dissolve the crude compound. Okay, so this is referred as recrystallization. So here the selection of solvent is playing an important role. Okay, you have to select the solvent in such a way that it will dissolve the product and not the impurities. It should have preferably the low boiling point. Okay, so suppose if you have the option of multiple solvents which are able to dissolve your crude derivative, then you can select the solvent in such a way that which will have a low boiling point, because once you get the filtrate you are supposed to evaporate the solvent so that the crystal will come out and you will get a solid derivative. Sometimes it is very difficult to get a satisfactory solvent which can dissolve selectively your solid derivative. So in that case what you can do, you can have option of mixed solvent for recrystallization. You can select more than two solvents so that you can dissolve the product, and the insoluble impurities can be retained on the filter paper. So this is how you can perform the recrystallization, and then the pure derivative is obtained, and this will give a very sharp melting point which will help us to identify the compound. Now we'll see how these derivatives can be prepared with suitable examples. So the following table is a list of some of the classes of organic compounds and a selection of derivatives that may be prepared to characterize them. Okay, so for example, on the left side we have a class of compound, and on the right side we have the name of derivatives which can be prepared from the respective compound. For example, alcohols. Now let's say you have given with the alcohols as an unknown compound. So alcohol you can convert it into its derivative, that is referred as 3,5-dinitrobenzoate. Similarly, if you have been given with the phenols, okay, you can convert them into a benzoate, acetate, bromo derivatives. Okay, then aldehydes and ketones you can convert into semicarbazone, then 2,4-dinitrophenylhydrazone, or you can convert it into oxime. Then acid can be converted into anilide, amide, and paratolidide. Okay, so these are the derivatives which are popularly prepared for acids. Then amines can be converted into benzoyl, acetyl, and sulfonamide derivatives and these are the solid derivatives, okay, of the corresponding classes of compounds. Okay, so in today's session I will tell you some representative examples, okay, where you can convert the unknown compound into its suitable solid derivative, and then you can find out the melting point of those. Okay, so for example, you can convert the alcohols to 3,5-dinitro-benzoate. Okay, so it involves a series of steps in the conversion. So what you have to do, first you have to heat gently a mixture of 3,5-dinitro-benzoic acid. So you can take 1 gram of this 3,5-dinitro-benzoic acid. You can see there are two nitro groups and there is a carboxylic acid. So it is 3,5-dinitro-benzoic acid is heated with phosphorus pentachloride. Okay, so there are 5 chlorine atoms connected to this phosphorus, so it is referred as phosphorus pentachloride. So 1 gram of 3,5-dinitro-benzoic acid and 1.5 gram of phosphorus pentachloride, you have to heat it gently in a dry test tube. Okay, and you have to heat it until it will liquefies. Okay, so it will take approximately 5 minutes to react, and then it will undergo the liquefaction. Okay, so this reaction you have to do it under the fume hood. So you know what is fume hood? Fume hood is a area which is a dedicated area which has the exhaust fan and the tube lighting, and whatever fumes are generated in the reaction, that will be taken care by the exhaust fan, and it will be sent out of the lab. Okay, and you have to take care of your eyes and nose, because the fumes which are generated, they are quite irritating. So preferably you have to do this reaction under the fume hood. Then once the mixture is liquefied, the liquid is poured on a dry watch glass and you have to allow it to solidify. Okay, so it will solidify. The phosphoryl chloride are removed by pressing the solid with a spatula on wad of filter paper. Okay, so this you can easily do it, and then you can remove the unreacted compounds. Then the residual acid chloride is suitable for immediate use in the preparation of derivatives. Okay, so what we are getting, you are getting the acid chloride. So what we have done, we have reacted 3,5-dinitrobenzoic acid with that of the phosphorus pentachloride, which have been converted into the residue of 3,5-dinitrobenzoyl chloride. So you can see the chlorine atom has attached here. Okay, so this OH has been replaced by the chlorine and it has formed the corresponding benzoyl chloride. Now this benzoyl chloride we have to take, and then this we have to react with the alcohol. So what is alcohol? Alcohol is our unknown organic compound. We have performed the preliminary test, elemental detection, solubility test, and we know that it is a alcoholic functionality by performing a functional group detection. And now we are supposed to convert this alcohol into its 3,5-dinitrobenzoate derivative. Okay, so the alcohol and the benzoyl chloride residue, now we are reacting together. Okay, so the benzoyl chloride is mixed with alcohol of 0.5 to 1 ml in loosely corked dry test tube and heated on steam bath for 10 minutes. Okay, so you know this alcohol, it can be a primary, secondary and tertiary. If it is a primary, you can heat it about for 10 minutes. If it is secondary or tertiary, then you have to heat it for about 30 minutes. Okay, so on cooling, you can add 10 ml of sodium hydrogen carbonate solution, stir until the ester crystallizes out and filter at the pump. Okay, using a Buckner funnel. So wash with little carbonate solution as a workup of the reaction, water, and you have to suck it unless it is dry. Then recrystallize from the minimum hot ethanol or light petroleum, and then you can get the solid derivative. Okay, so you have to make sure you have to cool slowly to avoid the formation of oily droplets of your ester. Okay, so there is a formation of ester here, and whenever you get ester, you have to be very careful because most of the esters, they are oily in nature. Okay, so this 3,5-dinitrobenzoate is a solid derivative. Only you have to take care or you have to be very careful when you are recrystallizing. So what we are doing here, we are converting our alcohol to its 3,5-dinitrobenzoate. Okay, so let's say it is a methyl alcohol, then here what it will form, this R represents the alkyl group of the alcohol, so it will form 3,5-dinitromethyl benzoate. Okay, like that. So whatever alcohol you have, whether it is a primary, secondary or tertiary, corresponding alcohol derivative in the form of 3,5-dinitrobenzoate will be formed, and this will have a very sharp melting point because it is solid compound and you can recrystallize, and because of that it will help you to identify the given unknown compound. Now let's see one more example where the unknown compound is phenol, and then this phenol will be converted to its corresponding benzoate. Okay, so you know what is phenol, it is a benzene ring and hydroxyl group. So to the phenol approximately we can take 0.5 gram, and this is added with 5% of sodium hydroxide as a strong base in a well corked boiling tube. Okay, so phenol and sodium hydroxide solution of 5% strength is taken, and it is corked in a boiling tube. Then benzoyl chloride of 2 ml is added in small quantities at a time, and mixture is shaken vigorously with occasional cooling under the tap or in the ice water. Because this reaction is exothermic, it will generate the heat, and to overcome this we can cool this reaction mixture under the tap, or we can provide the ice chest. Okay, so what it will do, by reacting the phenol with benzyl chloride in presence of this strong base, it will form the corresponding phenyl benzoate. Okay, you can see the phenyl benzoate has been formed here. So this H is removed, and then the benzene ring with this oxygen is getting attached here. So this will form a phenyl benzoate. After 15 minutes of this, you can get the solid benzoate which is getting separated out. The solution should be alkaline at the end of the reaction. If not alkaline, or if the product is oily, then you can add a solid pellet of the sodium hydroxide and you can shake it again. Collect the benzoate, wash thoroughly with cold water, and then you can recrystallize from alcohol or the light petroleum. So this is the procedure what you can follow for converting phenol into its corresponding benzoates. Okay, so again the phenyl benzoate is a solid in nature, and you can recrystallize it to get it in a pure form, and once it is pure you can find out the melting point by the procedure what we have discussed in the earlier videos, and then you can match that with the literature or the characteristic melting point of this phenyl benzoate. So this will definitely help to identify the compound identity in a very accurate way. So that's why this solid derivative preparation is very important in qualitative analysis. Okay, so hope you have understood the derivative preparation, why it is required, how it is to be done, and I have given an example of two compounds which you can convert it into a suitable solid derivative. So in the syllabus we are supposed to have or prepare only two solid derivatives, and there are other compounds also which you can convert into its corresponding derivatives. Okay, so I have listed here in this table aldehydes and ketones, acids and amines. These can also be converted into corresponding derivatives. Okay, so thank you for watching this video.